Well, we're very pleased to be able to welcome today Dr. Craig Young, who's going to speak to us and who's also been recognized as an honored alumnus for homecoming representing the College of Life Sciences. I'm happy also to welcome Craig's wife Robin, who's here with us, his parents, Martin and Gwen Young, his wife's mother Barbara Boyle, and three of the children I think are here. April, Andrew, Jessica. We're glad to have all of you here with us. We'll begin with an invocation by Will Winder.
Our Father in heaven, we're grateful to gather here as members of the College of Life Sciences. We're grateful for the many blessings we enjoy as faculty and students of this college and of this university, and the many blessings it brings into our lives. We are grateful this morning to have Dr. Young with us. We're grateful for his distinguished career and for the example that he has set for all of us, and for the impact that he's had in his field of endeavor. We pray that God bless him this morning, that he may express to us his thoughts, messages that will uplift and help us to do better. We pray that thy Spirit will be with us here, and thank thee for our blessings. In the name of Jesus Christ, amen.
Now, Dr. Young, if you wouldn't mind coming up here for a minute. This course is kind of short, so we're limited here. In addition to recognizing Dr. Young as our honored alumnus this year, we'd like to present him with something so that it'll be very easy for him to remember today, and it'll be very hard to think that this could possibly have come from another place. We have a former faculty member, Dan Fairbanks, who's also a sculptor, and we'd like to present you with this. You can pick it up if you want, but Dr. Lee Breathwaite, if he would come up and make the formal introduction, and we'll put this up all your time.
I'm very pleased we're going to get chairs. We don't have to do that in such meetings. Dr. Craig Young is a very special person. I've known him since he was an undergraduate, and as a graduate student, he's been blessed with superior abilities and gifts. He's used them very well. When he first came to BYU, he was a magician with the touring Cheney, is it Thompson? The entertainment crew. Craig was a true magician, never showed me a trick. I kept asking for one. Whatever he does very well. He was an excellent ideal student. And the very best student that I have taught. And I'm no spring chicken. I've seen a lot of students He is an excellent model for many things. Up front, he's a top-notch individual. He's a rheologist. He's been around spiny animals and so long that he's grown some on his chin. So you forgive him. He's just as good as you are, even with his spine. Craig is a wonderful researcher. I have never seen anyone that could get more out of research so quickly. I have been told that on a trip, a flight from Portland, Oregon from London, that Craig wrote 3 papers. He's a prolific writer in referee journals. How many to date? I lost track, about 150. 185. He's a model for all in academics. He's a gifted teacher. I asked him to give a couple of lectures just so I could sit and take notes. And his organization is supreme. His organization leads to wonderful things, one after another. He is excellent in his church work and in his family life. I have never seen anyone work harder. He has been at the Harbor Branch Oceanographic Institution for many years, deep sea research, so he is so qualified. I don't know if he will agree, but I think he's in the top 10 of deep sea marine biologists, not only in the States, but in the world. We are privileged to have such a superior person to deliver a message today. And I can promise you will be enthralled with his presentation. I turn the time over to you, Craig.
Thank you, Lee. That's the strangest introduction I've ever had to I've been introduced to a lot of seminars, and I've never before heard one that I couldn't believe anything of. In fact, Dean Brown, I'd never met Dean Brown before, but I'd read one of his books, and he called me up a couple months ago and said, we want you to come and give a lecture and receive an award. And the first thing that jumped into my mind was that he obviously didn't pull my transcripts and have a look at them. Because if he did, he would have seen those bad grades in various biology classes and my horrible grades in organic chemistry. So this is not the kind of role model that you really have to want. But I'm going to give you a lecture anyway, despite that. The theme of the homecoming, as you know, has to do with mountains and reaching the summit. So I'm having fun with that by talking about reaching down to the summit. We'll talk about mountains that are actually underwater. But before we do that, I want to tell you a little bit about Lee Braithwaite, because I knew he would try and embarrass me, so it's my goal to embarrass him just a little bit. This is a picture of the class that we had at Friday Harbor, Washington in 1973 on the first trip that I took with Lee to the Pacific Northwest to a marine lab. At that time, he was staying out there for a month. I think he does it for about 6 weeks now. Here's a few other pictures showing the good time that we had. What a tremendous opportunity it is for students to go with Lee Braithwaite to a marine laboratory and dig into research and learn from a master teacher. One of the things that we did on that particular trip was try and get the smoke going back down into the chimney of the girls' dorm, as you can see here. And I think that same trip we also walked a raccoon in the girls' dorm that emerged when they opened the door. And so here's a picture of Lee at the end of the trip on the ferry going home. My favorite picture of Lee Braithwaite, though, is this one. He knows his picture. I took this a year or two later during one of his field trips in the Rock Gator tidal zone on the San Juan Island. He might have made us wear hip waders, and he wandered around in his red socks taking pictures. Lee has been a tremendous influence on my life. All right, we'll talk about it, but one of the great highlights in my life was an opportunity to take Lee with me to the Bahamas quite a few years later. And there's a picture of us in Nassau. And to send him down in the front seat of a Johnston Sealink submersible with a 150-foot roll of film. He's a great photographer, he likes film. So sent him down with the camera and told him to take a picture of anything he wanted to. And then I sat in the back and interpreted things into his ear through a microphone. Subsequently, I was able to do the same thing for my father. He's here in the front row. And he's also one of the people, my whole family actually, that I owe a great debt of gratitude to. One of the most satisfying things I've ever done was have a chance to pay these folks back by sending them down on the adventure of a lifetime into the deep sea. So we're here to talk about mountains today. This is Mount St. Helens up in our neck of the woods. A little bit of smoke coming out of it, still an active volcano. What most of you probably don't realize, or don't fully realize, is that most of the mountains in the world are actually underwater, and that actually many of the biggest mountains in the world are underwater. I'm going to show you during the course of this talk a number of these kinds of diagrams, which are three-dimensional images that are created by sonar. This is a multi-beam image that shows the topography of a couple of underwater mountains or sea mounts. It's all done from a ship. As you can see there on the left, by bouncing sound waves off the bottom. Well, most people don't realize how many sea mounts or underwater mountains there are. In fact, scientists have just started realizing this in the last 2 or 3 years, last 10 years perhaps. We knew that there were thousands of sea mounts, but now with the modern technology that we have, we realize there's probably over 100,000 large mountains over 1,000 meters tall on the ocean floor. And there's a good chance that there could be 2 or 3 times that many. Very few of them have been explored at all, other than making those images remotely. Largest mountain range on Earth, of course, is the Mid-Oceanic Ridge. It runs right down the middle of the Atlantic Ocean and around all of the continents of the world. Here's a diagram here showing just how tall and impressive those mountains are. Somebody says that if you were to add up the Alps and the Rockies and the Andes all combined together, the Mid-Ocean Ridge would be about 4 times longer than those 3 major mountain ranges. And so by exploring mountains like Timpanogos, you're only getting the tip of the iceberg, so to speak. Here's a picture of me standing on the Mid-Atlantic Ridge. The Mid-Atlantic Ridge emerges from underwater at a certain point in Iceland, and there's lots of tectonic activity there, lots of volcanism and geysers and so on. And this is a rather bad picture of me with 1 foot on the North American tectonic plate and 1 foot on the European plate, and those plates are spreading apart, creating mountains on the ocean floor, and at least it's a slow spreading center, so I didn't fall into the hole. Now, if you'll indulge me for just a minute, I want to tell you how my master's thesis at BYU relates to what I'm going to talk to you about today, because I'm still working on the same stuff. My master's thesis was 96 pages long. It was full of all kinds of references and pictures and things, and I can sum it up for you in about 10 seconds. This is a sea squirt here that uses flow, uses water currents to help its feeding, and this is the larva of the sea squirt that selects where it has to live, and it lives next to its neighbors. Those were the 2 chapters of my master's thesis, and I labored over them mightily at the time, but I can sum them up that quickly. I'm going to talk to you for just a few minutes before we get into mountains about some of the problems of feeding in the deep sea. It's a challenging place to have to find food because the food is somewhat limited. I want to illustrate this at the outset by talking a little bit about sea squirts, or ascidians. These are 2 ascidians that are new species that I've discovered during the course of my career, and they have holes in them, an incurrent and exurrent siphon. And if you look inside of them, they have a branchial sac, which is has pharyngeal gill slits. You biology students know all about chordates. These are chordae, invertebrate chordates. They have pharyngeal gill slits, and those gill slits are lined with cilia, and the cilia drive water through so that the animals can collect food and so that they can respire. And the animal that I worked on, one of the animals I worked on for my master's thesis was this animal here, which is a sea squirt that stands on a tall stalk. It has an in-current siphon that's pointed down toward the base, and an ex-current siphon that points up. And the major discovery of my career so far took place on a dive in Monterey Bay, in which I was trying to fight the current and hold myself in place. Me and my buddy looked at this sea squirt that we were staring at, and as the current dented over, we noticed that that in-current siphon, pointed normally to the bottom of the animal, was pointed directly into the current. And as the surge shifted in the other direction and the animal moved to the other side, it was still pointed into the current. So by having a stalk, this is an animal that was taking advantage of the ambient water currents in order to facilitate its flow through internal feeding structures. Unbeknownst to me at the time, there are a great many animals in the deep sea that have stalks. I had no idea at the time, but this had been known since the Challenger expedition way back in 1872. Here are a couple of deep sea squirts that have very impressive stalks. And what you notice if you look at these is the in-current siphon is pointed down toward the base of the stalk, just like in that intertidal Stylomonariensis, montereensis, and the ex-current siphon is here. And the thing that really surprised me, and I discovered this in literature, is that these animals have branchial sacs that completely lack cilia. So this is an extreme situation. These have to use water currents. They have to use water currents in order to feed, or they wouldn't be able to feed at all, because they have no way of driving water through on their own. Well, the further we look, the more we find that in the deep sea, these kinds of things become really important. And so this idea of using stalks to facilitate orientation and feeding in filter feeding animals turns out to have great implications in the deep sea. I want to show you another species. This is another new species that we've recently discovered. This is a group of sea squirts called octavenids. It's about the size of a softball, I suppose. And these are carnivorous ascidians. They don't feed on plankton the way most ascidians do. I keep making mistakes there. They have great big wide open mouths. The in-current siphon goes all the way back and forth like that. And if you look at the branchial sac, it doesn't have any cilia either. It has no apparent way to move water through its system whatsoever. It has to rely on something else. These were discovered also at the time of the Challenger expedition. And at that time, this is what one looks like. At that time, actually much later than that time, I suppose, people noticed that it looks very much like Audrey 2 and the Little Shop of Horrors. And they assumed that these things probably feed like Venus flytraps. You find fish in them and crustaceans and so on in them instead of phytoplankton. And so people thought they'd probably sit there with their mouths open and then they slam them shut and gobble up these various little fishes and things. Well, we had the opportunity using a submarine to actually see how these things work. I'm going to show you a little piece of video footage here of one of these large ascidians. You can see here in a highlighted area that it has a fish captured inside of its body. That's the big wide open mouth down there. That's the in-current siphon. And the ex-current siphon is up here on the top. Okay? It has some strong muscles up on the top with which it can pump food once it closes its mouth back into the region of the esophagus. But we wanted to know how this animal captures food without any cilia. And so I convinced the submersible pilot, a very talented submersible pilot, into letting me put a Pasteur pipette on the end of a mechanical manipulator arm on the front of the $10 million submersible, and challenged him to using a hydraulic pump to filling up this ascidian with fluorescein dye. That was a challenge, but he rose to the challenge, and he injected dye in, and managed eventually, after a couple of tries, to fill this animal up with dye. So its whole in-current cavity here is now complete. We're measuring current with the current meter up here above. This is the ex-current siphon here, and now when it opens its mouth, what you can see is that because this siphon is higher than this big wide open siphon, and it's in the benthic boundary layer of the sea floor, Bernoulli's effect, remember that, students from physics? Bernoulli's effect causes a reduced pressure up here where the water's flowing faster, and so it pulls the water gently inside. And so the way this ascidian feeds, and many ascidians feed in the deep sea, is by positioning their body in a way that they can take advantage of the ambient water currents rather than having to expend energy on pumping themselves. They do it very, very efficiently by using the local currents. Well, as we start looking at other groups of animals, we find that stalks are incredibly common in the deep sea. Here are some in Hawaii showing several different kinds of cnidarian animals up on stalks so they can get up into the current. Here's a series of crinoids in the Bahamas. These are stalked crinoids. You can't quite see the stalks here. Maybe over here you can. But they stand up on a stalk, they hold a posture like an umbrella so that they can capture food as it's flowing into their arms. When animals don't have stalks in the deep sea and they need to access the foods in the currents, very often you find them up on pinnacles. And this is where we start getting into the concept of mountains and the importance of mountains. These are ophiocantha brittle stars that are filter-feeding brittle stars climbing up on the very top of the rock in order to feed. Animals that don't have their own stalks often climb up on animals that do have stalks. And so here we have a bunch of ophiuroids that are taking advantage of their neighbor, like Oregonian, in order to get up into the current. And here's another one, an Antipitherian here, with a whole bunch of brittle stars that climb right up to the very top. They're actually weighting down their neighbors to take advantage of them. Okay, so with that background about deep sea feeding biology, food is scarce, that's an important point. Animals want to save as much energy as they can, that's another important point. And the way that they do that is they get up high where the currents can be used in order to drive the foods through their bodies or around their bodies. I'm going to take you on a little bit of a tour here of 4 different summits or 4 different underwater mountains that we've worked on in my lab. One of them is Mauna Loa Volcano in Hawaii, which actually sticks up out of the water. Biosca Knoll, which is a small mountain in the Gulf of Mexico. We'll then talk about Lulu Seamount in American Samoa. And finally, I'll talk about mud volcanoes that are found throughout the North Atlantic Ocean, and then try and wrap it up in about a half hour's time. Here's a multi-beam map showing the islands of Hawaii. Hawaiian Islands, as you probably know, are slowly drifting to the northwest, and as they go over the hot spot, the magma chamber down below, whatever's over that is an active volcano. So we have these 2 very big volcanoes on the big island of Hawaii. This one is Mauna Loa. The largest one is Mauna Kea. And here's another image of Mauna Loa Volcano. The sites where we'll be working are right over here on the Kona coast. One thing you probably don't know about Mauna Loa and Mauna Kea is that they're the biggest mountains on Earth. Mauna Loa is by far the largest volcano on Earth in area. And if you consider the fact that Mauna Loa and Mauna Kea actually originate on an ocean floor which is a flat 5,000 meters on the bottom of the ocean, if you look at their entire elevation, they go up to 9,180 meters above the seafloor, and Mount Everest only has an elevation of 8,848 meters. And so you may think that Everest is the largest mountain. You may think it's also the most expensive mountain to climb. But if I told you how much those submersibles cost, you would realize that it's actually more expensive to do dives on Mauna Loa than it is to mount an Everest expedition, by quite a bit. So we'll be working on the Kona coast here in a couple of minutes, and some of you may have vacation in this area. It drops off very quickly, and so it's a nice place to get into the deep sea. I can have breakfast in a hotel in Kona, hop on a ship, and be at 2,000 meters in about an hour on the Kona coast, which makes it a very convenient place to do deep sea work. Submersible that we've been using in Hawaii is this one, Thesis submersible, Thesis 5. Which belongs to the Hawaii Undersea Research Lab at the University of Hawaii. And some of the animals that we see, not all of them that we see, but some of them that we see are sponges. And these are special kinds of sponges called hexactinellids, or glass sponges, that have skeletons that are quite firm that allow them to hold themselves up high in the water column. So although many of them don't have stalks, a lot of them can be a meter or even 2 meters in height because of those very impressive glass spicules that they have in their bodies. Let's just take you on a little bit of a dive here for a couple of minutes to give you some idea what it's like. This is the launch of a Thesis sub, or maybe the recovery of a Thesis sub, I'm not exactly sure. It's pulled in and out of the water on a big A-frame on the back of a large research ship. Inside the sub, you're sitting inside a 7-foot diameter titanium sphere and looking out 3 small portholes. And as you look out the portholes, these are some of the kinds of things that you can see. These are sea anemones here and sponges. Well, what I want to talk to you about specifically here is a quite remarkable finding that we made on the slope of Mauna Loa Volcano. And these are sponges. These are glass sponges, and they have a living part up on the top, and then they have spicules that hold up the sponge part up above the sea floor. And as we moved across the slope at this particular depth, we found that there were millions of these things. They're very, very abundant, undoubtedly the most abundant animals at that particular part of the sea floor. What I want you to notice here is that they have the same orientation and the same morphology, in a sense, as that little sea squirt that I worked on in Pete Braithwaite's lab way back in the 1970s. The ostial surface, which is the in-current surface, is formed into sort of a baseball glove. You can see it here. And the current goes in that direction because the stalk is flexible and the stalk is curved. So whichever direction the current comes from, it's able to reorient, and it's able to hold that in-current surface upstream. And the oscular surface, or the X-current, goes downstream. We tried to figure out why these things live in a very discrete band on the slope of the volcano. And in order to do that, we started looking at their feeding biology. One of the first things that's very obvious is that they're all oriented the same way. And we took current meters down and looked at prevailing currents. We also characterized the orientations of these things, and these arrows represent where the current would be coming from. And you can see that the promised beam payload, I wouldn't show any graphs, but I lied. I'm sure I'm showing you a couple. We have the same orientation essentially of all of these animals, and so they're taking advantage of the currents. Nobody knew what hexagonal sponges fed on. People had speculated about it, and so we figured out a way to determine that. And we built this little gadget here in the shop, and it's a gadget that has a syringe on each arm. And by holding it in a manipulator arm of the submersible here, and then going over with another submersible, another manipulator arm, and pulling a pin here, you can use the springs on the inside of those tubes to cause simultaneous water samples to be taken from wherever those two things happen to be. So we put one of them in the X-current flow of this animal to take a water sample, and we put another one out here on the side to sample the water that it might have experienced before it flowed through the sponge. What we determined was that these animals are feeding on heterotrophic bacteria. These are bacteriivores, which is really quite a remarkable thing. And so, trying to consider why they would be located in a discrete band, we did a series of transplant experiments in which we moved them up above and below the band where they live, and did controls in the middle, and then looked at various oceanographic parameters. And there's not time today to tell you the story, but we think we know why. It has to do with the fact that there's an oxygen minimum zone right below where that sponge zone lives. And so this is where the bacteria come to. It's where a lot of the bacteria die. It's where bacteria accumulate. It's a very, very good place for feeding. And in the transplants just below in the oxygen minimum zone, you can see still some of the dead stalks. And I'll let you read the paper instead of telling you the rest of the story. We have time now to dive on our second site. This is in the Gulf of Mexico. And the pictures that I'm about to show you, I took 2 weeks ago. We just came back from a cruise in the Gulf of Mexico, National Science Foundation supported research, in which we worked on cold methane seeps and on the various kinds of habitats that live here. So I'll show you first a few pictures of the muddy bottom, the flat bottom here, and I'm going to show you what a remarkable difference it is as we climb this peak, as we reach for the summit, so to speak, to use the BYU theme for homecoming. Down on the bottom of the ocean, down on the muddy sea floor, which is what most of the ocean is composed of, you can go for quite a ways without seeing too much life. A lot of the life is down below the surface. Here are 4 different animals, some fishes. This is a tripod fish standing up on its tripod on the stingray, for example. Here's a couple of other animals that live on the muddy sea floor. These are squid that hang out near the bottom. This is a sea cucumber that's able to swim up off the bottom that spends most of its time eating mud down on the seafloor. You find an animal here, you find an animal there. Here are a few more, a big lobster, a sea cucumber, so on and so forth. Now, I want you to consider what happens when we climb to the top of that peak. The top of the peak is a place where animals want to live. It's a place where animals want to live just like animals want to have stalks because they can get up into the currents. And so we have coral reefs up on the top of this hill. In the Gulf of Mexico. One of the largest deep water coral reefs that's known is on that hill. This is a deep water coral known as Lophelia. It's estimated that Lophelia worldwide covers probably 3 or 4 times as much area as all of the tropical coral reefs in the world. It's really probably the most abundant coral anywhere in the world. There are a couple of other pictures showing the Lophelia reefs. Lots of other animals live in the same area. There are a bunch of sea anemones. Here's a little Galatea crab living next to a black coral. Another species of coral. And I'd like you to look at this and tell me which way you think the current is coming from most of the time. It's coming from the left. These have formed little umbrellas in which they can capture the current that is coming from the left of the springs. Here's another Gorgonian that is up on the top, and you can see all these crabs are climbing up on the Gorgonian to get as high as they can, and then this is the conductor of the It's an absolutely spectacular sight. It's a riot of life. It's a place where there are lots and lots of species. Many of them are making their living from feeding in the currents. It's just a tremendous spot to dive. These are black corals here, by the way, Antipitherians, and some of these black corals are now known to be 2,000 to 3,000 years old. They're some of the oldest known organisms. They have lived in this place for a very, very long time. Another large black coral here, and lots of other animals. Well, you get the idea. When you get up to the top of the peak, ascend to the mountain, so to speak, reach for the summit, that's where the nourishment is located at, in the deep sea. A couple of reasons for this have been investigated by physical oceanographers. Not my work, but it's very clear what's going on here. We think of this cartoon as being the summit, and we think of these arrows as representing current that's flowing by. If you put a big mountain in the way, all of that flow has to go somewhere. It's either got to go around the edge or it's got to go over the top. And a lot of it goes over the top, and as it goes over the top, water's incompressible, so those streamlines have to get closer together, and that means that the water's going to be flowing faster across the top of the volcano. So in terms of the flux of food, or the amount of food that passes over per unit time, if you're up here, you get a lot more food in a given amount of time than you are if you're down here on the muddy bottom. That's just a very simple fact of physics. And so this is the place where animals in the deep sea choose to live. Another physical feature that is a consequence of the flow going past large mountains in the deep sea is that you get anti-cyclonic flows created over the top of these things. And so you get water that's flowing around in a circle and forming a sort of convection cell that right in the very middle carries food and current directly down to the top. So an animal that's sitting right here is going to have an opportunity to receive much more food than even an animal that's located on the flanks or the side of the volcano. Summit number 3, we might make it. Summit number 3 is in Samoa. And Samoa is a great place to go. If you're a member of the church, you'll find that about every other person you talk to is a member of the church, and they're all enthusiastic and smiling and great fun. Samoa, as you probably know, is a couple of different countries. The actual original country of Samoa, or Western Samoa, is located out here. These are volcanic islands that were formed as this chain, as the tectonic plate that this chain is on, moved over a hot spot, just like it did in Hawaii. This is American Samoa, American territory. This island right here is Tau Island, which is the easternmost island, large island of American Samoa. The place that I'm going to take you today on some submersible dives is this one known as Wailulu. Wailulu is an underwater mountain. It's a seamount. And it's a seamount that we didn't know existed 10 years ago. But there was a lot of tectonic activity here, a lot of seismic activity here. The geologists started looking, and they went out with multi-beam and discovered that this was the source of the volcano. This is the source of the earthquakes. And the reason is that this is the spot over the hot spot right now. Remember there were earthquakes just a couple of weeks ago in Samoa? This is where they're at. I'm going to take you to where those earthquakes are located. Well, this isn't just a little bump on the ocean floor. This is a mountain. You think of this ocean floor as being around 5,000 meters down here on the abyssal plain. The top of this thing rises up to about 600 meters below the surface of the water. And so if you've ever seen Mount Hood or Mount Shasta or Mount St. Helens or Mount Fuji or Mount Kilimanjaro, this is a mountain on the same scale as those kind of huge volcanoes. And we didn't even know it existed 10 years ago. Quite an amazing thing. So I worked with a geology team at Scripps Institution of Oceanography in San Diego, and my lab provided the biology team. We convinced the federal government to send 2 submersibles into the South Pacific for the first time. This was the Pisces 5, and we took the Pisces 4 with us as well. Here we are in Pago Pago Harbor, waiting to go out and have a look. Before I take you down on that volcano, I want to just give you an idea of what the local seafloor looks like. And so we'll make a dive first on an inactive volcano, Tau, which is an island on American Samoa. And the first thing that we'll see is pillow lava. These are lava that's extruded into that cold deep sea water and forms essentially strands, a big toothpaste of lava that hardens immediately when it hits the ocean floor, or hits the deep ocean water. And so we're going to run up Tau and look at some of the islands, fauna, down on the bottom of the ocean. These are pillow lavas. So that's what the volcano looks like when the volcano is first formed. What you see is lots of these animals that stand up off the bottom, getting high in the water so that they can get out of the current. You can see some current flowing from left to right there. And this is a crinoid, a natural crinoid that's using that current. A hexaconella sponge over here. Not very much life until you get up toward the top, where the most current goes. This is a fish known as a rat tail, probably the most common kind of fish that you'll see in the deep sea. And this is a deep sea fish called the Synaptobranchid, or cutthroat eel. Most of you won't be able to figure out what this is. I like to tell my students that this is an underwater elephant because you can see the trunk here and the ears here. This is actually a very common kind of deep sea cucumber, very gelatinous. These gelatinous sea cucumbers are so lightweight that they can actually pick up and fly off the bottom for ways. If you disturb them, they like to do this. Hermit crab with a zoantha on his back. Okay, it gives you an idea of some of the things that you might find. Well, this is a contour map showing by Lulu Volcano in 1999 when it was first discovered. What you see here is a crater, or a crater that surrounds a caldera. The caldera is about 1,000 meters deep. The edge of the volcano is about 5,000 meters deep. So we went down there with the intention of diving in this caldera for the first time and exploring and seeing what was there. We had no idea what to expect. And we got there in 2005, and as most of us were sleeping, the captain was running multi-beam surveys back and forth across the top of the volcano. And when we woke up in the morning at breakfast, he says, there's something that you guys really need to see before you make this dive. And it turned out to be a volcanic cone that had grown within the last 6 years. And subsequent isotope work done by our geology friends showed that this actually arose sometime about November, before the May we were here, just a few months before. And so this is a very active volcano. So we quickly talked the pilot into not going back to Honolulu, but into actually making a dive. My geology colleague made the first dive, and he landed right here on the edge of the crater, and then they looked around to see if it looked safe, snuck down to the ocean floor, and then climbed that volcanic cone. So it's an adventure. Science is an adventure. That's one of the messages I'd like to give you as students. To give you an idea of what this adventure was like, I don't know if any of you have been to Crater Lake in my state of Oregon. But this is Crater Lake. It's a volcanic caldera as well. It's now full of water. It's got a big rim. The bottom is about 1,000 meters deep, just like Lulu Volcano is. And it has something called Wizard Island, which is a crater, a volcanic crater that arose after the original caldera was formed. And so what we are looking at is the same kind of thing. Lulu Volcano looks just like Crater Lake. That's what we're exploring. I'm going to take you on a dive on the top of Lulu Seamount. This is an octopus that met us most of the time that we were there. A very large octopus. We're up on the top of the rim now, and then we're going to go down into the bottom and then climb up to the top of that caldera, of that volcanic cone. And here's what we found on the top of the cone. We immediately called it Eel City because it was completely covered with cutthroat eels. I didn't know quite what to do with this. I was an invertebrate zoologist prepared to work on hydrothermal vent animals here, and we found nothing on this volcanic cone but a bunch of eels. Well, started thinking about it from an oceanographic standpoint and trying to figure out what they might be doing there. And this is the top of the cone, Eel City, as it was called. A couple of pictures.
So as we tried to figure out what these animals were doing on top of that cone, why there was such a concentration of eels there, we put out current meters in some of the places where the current could flow through, the breaches, the lower places on the rim of the volcano. And these were some of the acoustic current meters that we put out there. And one of the things that we found that was interesting was that there is an anti-cyclonic flow. And so the direction of the current changes during the course of each day as we go through the day. And so there's flow that goes around in a circle on top of the volcano. If you remember the diagram I showed you earlier about how the water flows around in a circle on the top of the volcano, we concluded that this is forming a Taylor's column that's actually delivering food right down to the top of the volcano, right where that volcanic cone is located, where all those eels were hanging out. And so what were the eels doing? We think they're probably taking advantage of the food on the top. So we tried catching them. We had a second expedition there a few months later. We took out various kinds of traps, baited them with things that ought to be really tasty to eels, and we didn't catch a single eel because we're a bunch of invertebrate zoologists and not fishermen. We didn't know how to do it. And so we went into the ship's shop and we manufactured a butterfly net that a very talented pilot could use with this mechanical manipulator arm. These guys trained by playing video games. And he caught probably 60 or so eels with this butterfly net. And then we discovered that the eels are really good at worming out of the net backwards by their tail. And so we ultimately came up with 7 eels after an entire cruise. And this is one of the eels. It turned out to be a species that was already known, that had accumulated in great numbers here since that volcanic cone had formed. We looked at the insides of the guts and found that what they're feeding on is midwater crustaceans, such as shrimps and euthausids and things like that. These are animals that are found in the water column quite sparsely, but where the Taylor's column concentrates the flow, they're found in great numbers. And so they have a perfect place to live at the very top of the summit. We also found hydrothermal vents, chimneys around part of this volcano as we explored it. And then as we went down to the very bottom of the volcano at 1,000 meters, what we found was a stark contrast between what was up on the top and what was on the bottom. Everything on the bottom of the caldera pretty much was dead. These are all dead fishes, dead squids. The whole thing was entirely littered with the carcasses of animals from midwater who had gone down into the depths at the base of the volcano and perished. So I'll take you down there. This is sort of a Hades situation. Brimstone everywhere, sulfur, and you get the picture. And down on the bottom, you have microbial mats and you have bubbles coming out of the sea floor. And as we sampled the bubbles and we sampled the seawater, we discovered this is carbon dioxide. Carbon dioxide naturally bubbling in great amounts out of the ocean floor from this volcano. And everything was dead there because it was pH 2, except for this animal here. There was a little red worm full of hemoglobin that was able to feed on the carcasses of the fishes down here in this area that we call the mode of death. We don't know what this animal does. Here's a picture of it in the laboratory. We captured 3 or 4 of them. A good physiologist needs really to work on this and discover how it tolerates pH of 2 and those tremendously adverse conditions down in the bottom of the caldera. I'm almost out of time, but I'm gonna give you the last little story. This is a mud volcano that I photographed just 2 weeks ago in the Gulf of Mexico. These are found throughout the world in places where methane seeps out of the ocean floor. And other kinds of similar circumstances where methane seeps include underwater lakes. This is an underwater lake here called the Brine Pool. Here's a little bit of video footage that was taken by the people who did the Discovery Channel Blue Planet. They filmed a good part of that on my cruises, if any of you saw that on TV. So here we are cruising over an underwater lake that has a salinity of 120 parts per thousand that doesn't mix with the overlying salinity of 32 parts per thousand, so it maintains itself as an underwater lake, and it's surrounded by mussels. These mussels all the way around the edge of the pool are feeding on methane, using bacteria located in their gills. They're taking advantage of the methane gas that's bubbling out. They don't need to feed except to give a little bit of nitrogen into their systems because they have a continuous and beautiful source of carbon in the form of these bubbling methane seeps. Okay, this is a close-up picture of what they look like. This community supports all kinds of other animals, including little shrimps and little snails and so on. Now, the last little bit of the story that I want to tell you has to do with not feeding, but the larval biology of these animals. It's recently been discovered that these mussels that live in the Gulf of Mexico either live or have close relatives that live at cold seeps all the way around the Atlantic Ocean, including off West Africa in the Gulf of Cadiz. And there appears to be, genetically, there appears to be some continuity between them. And it's been, we've speculated about how they might be able to do this, because the currents down in deep water where these animals live go so slowly that if an animal were to try and disperse as a larva in those deep water currents, it would take 5 or 6 years. No larvae are known to be able to do that, and they get lost or eaten probably on the way. There are surface currents in the tropics in the Atlantic Ocean now, moving east to west, right in the middle, and west to east, north and south of the equator, that could get a larva across in probably a little less than a year. And so the question is whether these mussels and the other animals that live with them might be able to actually get up and tolerate the conditions way up in the surface waters in order to disperse these tremendously long distances. And so graduate students in my laboratory working with me on this have worked out the embryology of various cold seep and hydrothermal vent species with the view of looking at how an animal on one mountain can locate another mountain on the other side of the ocean where it might want to live. This is the embryology of that deep sea mussel right here. We put traps on the ocean floor to catch the larvae right as they seek their settlement site at the end of their larval life. And we were very successful in catching the little larvae as they came moving into the bottom of the ocean. We also used sampling device called a MOC-NES, multiple opening closing net environmental sampling system, the MOC-NES, which has a series of nets that can be opened and closed remotely from a cable on the ship. And here's a picture of me and one of my students pulling one of the MOC-NES nets over the side of the ship. We can sample discrete layers of the water column and figure out where the larvae are located. What we found out was that we can find Bathymodeolus larvae 500 meters above the bottom. This is an old slide. We've now found them at the top 100 meters of the water column. And this has been confirmed with genetics, so we know that these really are the right animals. Another species, similar kind of story, is this little snail that lives on the mussels. This snail is called Bathyneurida matacoidea. Cute little animals. They lay egg masses on the mussel shells. They hatch out of these egg capsules in about 4 months, and they hatch at a particular time of year between March and July. We did the embryology, looked at the larval stage, which is called a velager, and then looked for it up in the water column. This is what, well, look over here. This is a scale bar. Everything on this picture is going to be 500 microns. That's the 500 micron scale bar. This is the size that they hatch at, and we captured these things in traps on the bottom as they settled as well. And they're this size right here when they settle. And so they have to grow between that size and that size in the water column while they're swimming around as larvae. We found them in tube traps right there. And we looked at them genetically to make sure they're the right guys. And then we found them in November and February. In February, we found them 100 meters, from 0 to 100 meters below the surface of the water, about the same size as these. Confirmed them with genetics, sequencing, that this is the same animal. So now we know, these are 2 pictures of the shell. We now know that these animals are capable of getting up where the currents flow quickly and probably disperse from mountaintop to mountaintop in the deep sea by going way up to the surface and then going back down again when it's time for them to settle. That's a picture of one of the little larvae. Well, that's the end of my story, but if you'll let me tell another little special experience right at the very end, I appreciate it. A few years ago, another dean, who also didn't check my transcripts, offered me a job here at BYU in the zoology department. And I turned him down. And Robin, we were living in Florida at the time, working at a Mid-Oceanic Ridge institution. Robin and I came, and we looked at the situation. Neither of us felt very good about the situation. We thought it might not be quite right for us. We went to the temple. We saw inspiration about it. We turned down BYU. That's hard, to turn down Lord's University for a faculty job.
with an attractive salary. But we did. A couple of years later, we were offered a job at the Oregon Institute of Marine Biology at the University of Oregon, and we took it. No good reason. The following year, Stanford University, where Lee Braithwaite had been taking his remarkable field courses for many years, decided that they had to renovate the building that he had been going to. They didn't have a place for him to go. And so I talked Lee into coming to the Marine Lab that I now directed in Oregon and bringing his classes there. And it turned out to be a good situation. They've come back now for about the last 6 years. The first year that they came, I was sitting in Sacramento meeting, and it was fast and testimony meeting, and the BYU kids, 30 or so of them, showed up, and one by one they all went up and bore the testimony and expressed gratitude to Lee Braithwaite for providing this wonderful experience. And I knew right then that the reason that we went to Oregon instead of BYU was because BYU needed a place to go in Oregon. And I needed to be in a position to make that happen. So we appreciate BYU coming there. BYU Idaho also comes there now, by the way. And it's been a great experience to interact with BYU students, even though we're not at BYU. Thank you very much.